This Week in Virology, the podcast about viruses, the kind that make you sick. From the U.S. Army Medical Research Institute of Infectious Diseases in Fort Detrick, Maryland, this is TWiV, This Week in Virology, episode 1015, recorded on June 6, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here at Fort Detrick, Rich Condit.
Hi, Vincent. Here we are on the road again. Are you recording, by the way?
Of course I'm recording.
Sometimes that doesn't happen. Not very frequently. At any rate, and I can't report on the weather.
I can't either. I don't have my phone. And
don't have my phone?
No phone.
You know? And we've been inside the whole time. But when I got up, it was a nice day.
It's a nice day out, yes. You confirm it's a nice day out, listeners? By the way, we have a fabulous audience of thousands of people. It's a huge room, and they're all apparently TWiV listeners. Today's a special day, right?
Yeah. Today is D-Day, the 79th
It's the anniversary of D-Day, right? Yeah.
79th anniversary.
Yeah, it's a pretty interesting day to have picked to come here to do that. So we're recognizing that. And so our goal is to give our listeners an idea of what's going on here. Because frankly, I didn't really know very much about it. We had a tour this morning where we learned a lot. And we have 4 individuals who work here. We're going to talk to them about what's done here and why it matters. And so let me start all the way on my right. The person who had actually invited us a year ago. Took a year to get us here. Todd Bell, welcome to TWiV.
Thank you for being here, guys. It's really a privilege and honor to have you here.
My pleasure. And then we're gonna do this alphabetically. So over to my left, Crystal Burke, welcome.
Thank you, and I'm happy to have you guys here.
Yeah, thanks a lot. Over on the left, John Dye, welcome to TWiV.
Yes, we're very excited to have you. Thank you for coming.
I should say welcome back because you were on once before, right?
Yes, sir. I was on in New York with the Prometheus team and Kartik Chandran talking about our monoclonal antibody discovery effort. That was about 6 years ago, maybe? I don't know what episode.
That was, I wrote it down here, but I deleted it, sorry.
It's the story of my life.
Do you know why I brought John back? Because he calls me Sir all the time. And nobody else does that, so I kind of liked it. I'm just kidding. And finally, here on my right, Jay Hooper. Welcome to TWiV.
Yeah, it's great you guys are here. I think it's the first podcast ever out of USAMRIID. So.
Is that right?
Yeah.
So that's a first for you listeners. It's not all secretive here. In fact, everything here is, what did you call it? Open
Unclassified research.
Unclassified research, so what is classified research? You can't talk about it, right?
Well, John, do you want to take that?
So we could tell you. Sure.
I tell you, but you have to kill you.
Classified research, we could tell you about it, but then we'd have to kill you on the way out. All the work that we do here is open source research. So we apply for funding just like any other organization, from NIH, from other funding agencies. And then just like when you sign a contract to do that type of work, you actually are saying you're going to publish in open source journals. So everything we do here thus far is unclassified, and we publish in all the same journals that all your listeners do. Hopefully Science and Nature if we're lucky enough.
All right. Well, we'll hear a lot more about that, but first I would like to hear from each of you a little bit about your training, what you did to get here. So let's start with you, Todd. Where are you from originally?
So yeah, I grew up on a farm in southeast Iowa, a farm kid, and initially wanted to be a veterinarian, and ultimately became a veterinarian. But when I was in vet school, my plan was to go back and be a vet, kind of a large animal vet in small town Iowa. But my mentor at the time, who was a veterinarian, kind of approached me when I was in vet school and said, I know you want to go back and work with guys like your dad and your uncles who are still small farmers in Iowa, but the ag community is changing. You're going to have the same amount of business, but 20% of the clients. The guys like your dad are about 20% of the folks, and 80% of our business is consulting with very large farms. And if you want to do that, that's great, but I just want to make sure that you're aware of that. And I thought about that, and I was like, I don't know that that's necessarily what I want to do when I grow up. And so I started looking at other routes and got picked up to be a summer student while I was in vet school at the pathology department at the National Zoo. And while I was there, some folks from the old Walter Reed's Armed Forces Institute of Pathology, now known as the Joint Pathology Center, they would come over and train with us at the National Zoo. And there was an Army guy there that made it sound great. And I still wasn't convinced though. But the gentleman at the National Zoo, Dick Montali, a close friend who has since passed, he knew the head of the pathology department here at USAMRIID by the name of Tom Larson, who was also a fellow Cyclone. And so he said, why don't you go up there and check out USAMRIID for a day? This would have been in the summer of 2000. So I walked around here that day with the head of the pathology department as a little veterinary student at the time and thought, this place is amazing. I want to go back to this magical place known as USAMRIID that does all this cool research, but I still wasn't sold on being in the Army. I thought I'd come back as a civilian maybe, right? And then I talked to a friend who was in veterinary school a few years ahead of me who joined the Army and made it sound great. And I had a high regard for Jennifer Chapman, who's since retired. And then right after that, 9/11 happened. And I thought to myself, well, you know what? I'm going to raise my hand and sign up. Even though I have no military training, I had no desire to be in. If they need veterinarians, I want to do this, and hopefully I'll end up at USAMRIID. And that was 21 years ago, almost to the day that I came into the Army.
So I want to, for the listeners, not necessarily these listeners, but the ones out there in digital land, USAMRIID. Have we said what that stands for?
Yeah, you were asleep.
I was asleep. Okay. So
Say it again.
You could say it
Say it
again.
again.
Yeah, so USAMRIID, the U.S. Army's Medical Research Institute for Infectious Diseases, or otherwise known as RID. People just shorten it and call it RID oftentimes.
Okay. And are we going to do we've got to do some history on this. Is now a good
Well,
time?
let's go
Introduce
through.
the-
Let's go through the people. You haven't been around for a while, have you, Rich? Someone else entered the services because they were a veterinarian that we've had on TWiV before. They said, and I said, why would you do that? And they said, well, this was a while ago. They had a lot of horses
They did.
to take care
Yep.
of. Is this still the case?
And they still do. So when I came in, my first assignment was at Andrews Air Force Base in Washington, D.C., where you generally, your first couple of assignments as a veterinarian, you're a general practitioner. So you have veterinary clinics you're in charge of, and then you take care of the military working dogs that are on those installations, then any privately owned pets that service members have, you take care of them as well. That being said, there are still horses at Arlington National Cemetery, and right outside of Washington, D.C. And I was the only veterinarian stationed here at the time that grew up on a farm, had any large animal experience. So they said, Bell, you're up. You're the horse guy. So you're going to take care of the horses at Arlington. So I did that the second year that I was here in the Washington, D.C. area. And then and then from there, you take one assignment in the U.S. and then you go overseas. I was with a deployable infantry unit, Special Forces unit out of Colorado, where I deployed with them to Iraq. And you kind of take care of locals' animals to kind of build relationships and trust that way. And then if you stay in the Army as a veterinarian, they force you to specialize. And so what I wanted to do is be a pathologist. I really enjoyed pathology in veterinary school and had a lot of great mentors at Iowa State. And so I chose that path and again went to the AFIP, the old Armed Forces Institute of Pathology. Did my residency there, and then 2010, that's when I came to USAMRIID the first time. This is my third time here. As a pathologist, and I just absolutely loved it. Got to work with, in a given week, you could work with Nipah, Lassa, hantaviruses, anthrax. The next week you'd be working on plague. It was, for a pathologist, it's like being a kid in a candy store here. It was wonderful.
So all the work we'll talk about, you've been involved as a pathologist, correct? Okay.
Yes, and then subsequent to that as a virologist as well. I enjoyed my first time here so much that the Army let me go back and get a PhD in virology at George Mason. They have a regional biocontainment lab not too far. So I left here in 2014, went to George Mason for 3 years in their regional biocontainment lab, got my PhD working with Rift Valley fever virus in Kylene Kenhall's lab, amazing scientist. And then came back here a second time for a year to be the military deputy to the science director. Left for a couple years to go to Japan to be in charge of the public health activity there in Japan, taking care of the vet assets and some food inspection and public health things, and then came back here in 2020 in July to take over foundational sciences, which John and I are in charge of. John and I are basically we get the honor and privilege of essentially kind of day-to-day operations of the science that happens here, which is our 4 science divisions: diagnostics, molecular biology, bacteriology, and virology. It's about 220 folks, equally split, kind of military, civilian, and contract workforce.
And despite being like 30 years old, you're retiring, right?
Well, thank you, Vincent. Yeah, my hair doesn't say I'm 30, but yeah. So I actually just had a retirement ceremony. I've seen a lot of people with quizzical looks on their faces here today. So I just finished 21 years in May, because I came in May of 2002, and finished 21 years. And after 20 years in the Army, you can retire, full retirement, and then transition into your civilian life. And so that's what I had a retirement ceremony on May 5th, but the Army has a great program called the Career Skills Program, where they give you a month or two to kind of go back to a civilian institution and kind of relearn your civilian skills a little bit before officially retiring. So I don't officially retire until June 23rd. And so back in an official capacity today, I had again, just a privilege to be here today, so I got to put the uniform back on for a day and come back, because I'm still technically on active duty, but my Career Skills Program, which ends this Friday, is at George Mason University. So I talked to the folks that I was a PhD in, Dr. Ginny Espina, and she let me come hang out for 6 weeks and just run assays in her lab to figure out which end of the pipette actually worked. You know, being a science administrator as a colonel over the last 3 years, I didn't have a lot of bench time. I still would go into the suites to do inspections and things like that, but I didn't have a lot of bench time. So.
And what are you doing next?
So next, I, again, very fortunate and blessed, Iowa State University, where I went to undergraduate veterinary school. They offered me a job as a professor in their pathology department. So they'll let me have my own lab and train their PhD pathology folks, and then I'll teach 20% of the time. But hope, you know, it's wonderful kind of growing up in this environment. I kind of feel like I'm a piece of this place, for better or worse, and it's just been a kind of a privilege and honor to serve the folks here and to lead the science. You know, walking into a room every day, understanding you're the least intelligent person in that room is the place I've been the last 3 years with all these brilliant people you work with. But John and I's goal really is the leaders is to give our folks the people, the equipment, and the command intent that they need in order to succeed, and then cut them loose, right? In the military, you have kind of left and right boundaries you got to follow, but we try to give them that. But good science is you know, thoughtful. It's discovery. It's kind of getting after things in a collaborative and open way, and we try to kind of set the groundwork for that over time, and thankfully our folks have really flourished in that regard. And I'm sure we'll talk about a lot of the great things that we've done, but most of that's been done in spite of us, not because of us, and we just try to stay out of the way and help our folks do good work.
Yeah, I want to have you tell us about the structure of this,
Sure.
this facility and how it works, but let's finish with some of our other guests. Jay, tell us your history. Where are you from?
Okay, I'm from Maine. I lived in a bunch of other places, but kind of from 6th grade on, I was in Maine. I went to undergrad at Colby College up in Waterville, Maine. I was a bio major, liberal arts degree, and up in Maine, coming out with as a bio major, there weren't really any, not a lot of jobs. And so I was looking around. It's really ironic. I happened to be in Portland, Maine. I went to the recruiter thinking, hey, you know, maybe I'd go military. And I was like, nah, this isn't for me. And it just so happened that there were some sergeants from USAMRIID who had been sent north, going up through colleges and other recruiting stations up the East Coast, and they happened to be there that day. And so the recruiter said, hey, there's some guys over here that are sciencey, doing science stuff. Maybe you should talk to them. So I went and talked to them, and they're like, yeah, you know, we work at this place called USAMRIID. It's awesome, virology. We can call up the lab right now, and you can talk to a scientist. So they did, and I'm like, yeah, maybe I can go into the military, serve my country, and see if I like research. You know, what else am I gonna do? And so signed up, and it was cool. I went to basic training, and at the time, the program was called O1H program, which means if you had a college degree and you went in, you'd be guaranteed to go to a lab for 3 years, and that was it. It was a guarantee. And they could pay off your school loans, or you can get the GI Bill. So it was pretty good deal. But it was definitely a gamble. So I ended up here. Todd said he feels like he grew up at USAMRIID. USAMRIID was 17 years old when I came here. And I was, so that was in '86. And so I was in the lab of Alan Smalljohn working on pox. Thought it was really cool. I did my 6 years, thought it was awesome. And then I was trying to figure out if I go to medical school or graduate school. So I applied to both, and I ended up, I got into Harvard, and which was pretty awesome. My friends in the barracks really were quite shocked. And so I left USAMRIID in '89, went up to grad school up at Harvard. I was in Bernie Field's lab. I was his last graduate student. So I was up there for 6 years. And then postdoc time, where do I go? So I was looking around, and I knew about USAMRIID, and I'm looking at other places, and I thought, I'd been doing academic reovirus, research. I'm like, I want to do virology that really has some application to it. And so I decided to do a postdoc at USAMRIID, maybe a year, maybe a couple of years. So I came here, I ended up in the lab of Connie Smalljohn, trying to do hantavirus reverse genetics. And so that postdoc was an NRC, so a National Research Council postdoc. That lasted about 3 years, and then I was brought on as a civilian, GS civilian, and then they kind of locked me in here, and I've been here ever since.
We locked them in.
So the first time you were here, you were military,
I was military,
correct?
yeah.
And then you left after 6
left.
years, okay.
Totally. People asked if the grad school was related to the military. It wasn't. It was totally separate. But then I did decide to come back here. And again, Todd mentioned a magical place. It is a magical place for this kind of research that we do. It's really amazing, and it has huge potential here.
Magical is what they call Disney World.
Maybe that's not the right word.
That's coming up with a word, maybe.
Yeah, so that's my life story.
Crystal, what's your story? Where are you from?
I am from the middle of Florida. So I grew up literally in Middleburg, Florida. I was not being facetious or anything. I did my undergrad at Jacksonville University in Jacksonville. From there, I went to Louisiana State University Health Science Center in Shreveport, Louisiana. And everybody has some sort of connection with military. The only connection I can say is Dr. O'Callahan, who was the head of that program, always told us graduate students that he wanted a few good Marines, not a bunch of Coast Guards. So I guess I was one of the few good Marines in that program. I did my PhD work under the direction of William Klimstra, which him and Kate Ryman were a husband-wife pair doing alpha virology and some flavies on the side, because you gotta spice it up a little. And I spent a lot of time understanding innate immune signaling and how the cell recognized viral infection and kicked off that whole interferon response. And when I say a lot of time. And so when I went to do my postdoc, I was like, A, I'm sick of alpha viruses, and B, I don't want to do cell signaling. So I was looking around, and I went to the NIH for their graduate or postdoc recruitment program, and I happened across a program at St. Jude Children's Research Hospital. And I went and visited with a couple of the scientists there and decided to go into Charlie Russell's lab, which was completely different because he is a physical biochemist and was interested in fusion proteins, which I was not. However, one of the side projects he had was to try to understand the pathogenesis of Sendai virus in its native mouse model. And before I had gotten there, they had made quite a few varieties of luciferase expressing viruses, which in 2009 when I started was relatively novel, especially for a negative strand virus, because they're a little more complicated than the alpha viruses, which had done that a few years ago. And so I took a project on to look at paramyxovirus pathogenesis. I morphed it into paramyxovirus transmission because I wanted to know what virus went and how. And was it contact transmission? Was it aerosol? Got a lot of experience modeling, and then had to decide, what do I want to do when I grow up, right? That's everybody's question. Having experience with a lot of in vitro cell modeling and mouse modeling, and having the St. Jude mission always in mind made me want to find a place where there was a purpose for what you were doing. And USAMRIID allowed that opportunity because we had a very specific mission, which is develop, medical countermeasures to help the warfighter, and hopefully which then could be used for the public health in general. And so Dr. Glass, Pamela Glass, who is an alpha virologist, which, as we always say, virology is a small world, she was actually in William Klimster's lab for a month when I first got into graduate school, and so I had some previous experience with her. She was looking for a viral vaccinologist. It was a very sexy sounding title. It was a contract position in the lab, so it's a PI, and she needed help with her project that she was working with the NIH to look at a vaccine platform that they had developed for the alpha viruses. And so I came here in 2014 to do advanced development of the virus-like particle vaccine platform collaboration, and I've been here ever since. About a year ago, she moved out of the lab and into biosafety, and so I took over the viral pathogenesis branch and have been hopefully moving us in a good direction and expanding from alpha viruses into a few other
There she is.
different families. Yay!
She trained us this morning.
Yeah.
Managing some biosafety or trying to, and it was time for her to take that rank.
Cool.
Well,
All right, so now you're on your own.
now I'm on my own.
You have support.
Okay.
I hope I'm doing everybody proud.
All right, John, tell us your story. Where are you from?
So I'm from nowhere. I grew up in a military family, so every 2 to 3 years we would move somewhere new. My dad was in the Air Force for 23 years, retired as an 06. So we moved, being on a military base is very normal for me. Listening to the morning revelry and then having taps at the end of the night is something I grew up with. My middle school and high school years I spent in Northern Virginia because my father was stationed at the Pentagon for his last station. So I was there growing up, and I knew I really liked biology, and I attended the College of William and Mary in Williamsburg, Virginia, and received a liberal arts degree with a biology major, much the same Jay did. And much the same as Jay, I had no idea what I wanted to do with that after I got out of school. So I moved back to the Northern Virginia area, and I'm going to date myself now because I actually went to the Chamber of Commerce for the local areas in my surrounding area and asked for a list of biotech companies, literal addresses of biotech companies, and I sent out mass resumes and CVs to 100 different biotech companies in the DC area. And I had 3 callbacks, interestingly. One was a place where I could do paternity testing. I didn't like that very much because I was worried I'd have to do that for myself one day. The second was actually a laboratory called Hazleton Laboratories, which is kind of interesting because there's actually a parallel to here at USAMRIID. I was actually interviewed to be an animal technician because everybody starts off in the animal realm, actually cleaning monkey cages in Hazleton Laboratories. And for those of you who don't know Hazleton Laboratories, that is where Ebola virus Reston started. So literally, I turned down that job, but Reston had not been there. But while I was working at this other job, that's when Ebola virus Reston actually came about. So I could have actually been at those laboratories in that time. The third place I interviewed was a place called SRA Technologies in Rockville, Maryland, and they did HIV research. We were actually a part of a team that did the GLP, and I know it's funny that I did GLP work back then, GLP work to test a non-nucleoside reverse transcriptase inhibitor called delaverdine. It was a product by Upjohn, which then became GSK, which then became a bunch of other companies. It's now part of the HIV cocktail. So we actually were testing those, doing syncytia assays, P24 ELISAs, viral burden assays, those kind of things. And I met there a Colonel Chet Roberts, retired Colonel Chet Roberts. He was my supervisor there. And he looked at me and he said, well, you're moving up pretty well. You're doing some middle management, but if you really want to make a difference in this world, you need to go back to school and get your PhD. So I applied to schools, and I started school at Loyola University Chicago for a PhD in microbiology and immunology. So I got really good at eating ramen noodle and peanut butter and jelly sandwiches in Chicago. And after 5 years, I completed my degree there. I was working in the lab of Dr. Dan Quinn. So he's an immunologist. So I'm actually an immunologist. Ooh, hiss, I know. Not a virologist by training, but we were interested in the effects of lymphocytic choriomeningitis virus on mice and the trafficking of immune cells after that infection. So it's actually a model for multiple sclerosis, where if you infect the mouse in the brain with LCMV, you get a mass infiltration of CD8 and CD4 T cells into the brain, which cause destruction of the brain, which is very similar to what occurs in multiple sclerosis, where you get a mass infiltration of T cells into the brain, and it's actually the immunopathology of those T cells that causes the issues in MS. After that, I was at a meeting in Colorado, and I was doing a lot of flow cytometry. Back then, it was a 5-color flow cytometry, just to give you an idea how long ago it was. Now there's 27, 35 color flow cytometry, but 5 colors, and I was like an anomaly to be able to do 5 colors, because most people were just doing 3. Dr. Mary Kate Hart, who is a PI here at USAMRIID, it was at a American Association of Immunologists meeting in Denver, noticed my poster, and she said, we have a bunch of vaccines for Ebola virus, and we need to look at correlates of immunity, and I want to bring this technology of flow cytometry into our laboratory. So she recruited me. I came for an interview, and I absolutely fell in love with the place. I knew I always wanted to serve my country. However, I also knew I was not going to be a very good soldier if I actually joined the military, because they don't take orders very well. But this was a great opportunity for me to meet that need to serve my country in a way that I could. So I came here in 2003. I started as a postdoc in her laboratory. In 2007, I became a PI, and then I've moved my way up through branch chief up to the deputy director of foundational sciences with Colonel Bell. And I absolutely love USAMRIID. It is in my blood, and it will be. I don't ever plan on leaving this place until I retire.
So you were on TWiV 578. So this is 1015.
500 TWiVs ago.
Well, that was towards the end of 2019.
2019.
So
It was before COVID.
It was just before COVID, and we started doing 3 a week, so that's why. But you were part of this Prometheus team making monoclonal antibodies, and I was curious, and we'll find out what you did during COVID with that technology. So we'll get back to you, but I want to go back to Todd, and maybe you could give us a sense of what USAMRIID, I keep saying it wrong.
Yep,
sorry.
USAMRIID. Mhm.
USAMRIID.
Or you can just say RID.
I like USAMRIID, that's what I'm used to. But what is it, and why do some of you have uniforms and others do not?
No, that's a great question. So USAMRIID's mission, and the military's mission in general, is readiness, right? So we need to be ready. And USAMRIID is the lead kind of medical biodefense lab for the military. And in particular, our readiness is kind of twofold focused. One is making sure that our soldiers are ready should they be deployed anywhere in the world, that we have a vaccine, a therapeutic, or a diagnostic in their kit bag for whatever region we send them to where something is endemic, right? And then the second part of readiness is being ready for an adversary should they have a bioweapon, or being ready for an emerging pathogen, right? That's been the majority of the threat over the last 10 to 20 years, has been emerging viruses. As population and travel increase, right, we're seeing these emerging viruses. And so as a military, we have to be ready to respond to emerging infectious diseases. So that's, USAMRIID's mission is really to be ready for those eventualities, right? And so again, we're defensive focused, and we develop vaccines, therapeutics, and diagnostics for the warfighter. Now, we're one of several military labs that have different focuses, right? So our higher headquarters, which is MRDC, Medical Research and Development Command, they're across the street. But we're one of a series of labs, and our focus is biodefense. There's also a lab focused on chemical defense in Aberdeen Proving Grounds. There's also a lab in Silver Spring, the Walter Reed Army Institute of Research. Their focus is a little bit different, wherein they do diarrheal diseases, they do malaria, they do kind of more endemic type things. They're not on the select agent list and not in BSL 3 and 4. So that's kind of where we have some convergence, but we also have kind of different swim lanes in that regard. And then there are other labs as well that do environmental medicine, that do other things for kind of soldier health and soldier readiness per se. But USAMRIID is one of those labs in our MRDC, with our focus being biodefense, and we are the sole biosafety level 4 lab in the Department of Defense.
So why the civilian military mixture?
Sorry, that's a great question. So USAMRIID, which you'll see on our website, it fluctuates around 650 individuals. It's about 1/3 military. It's about 1/3 civilian workforce and about 1/3 contractor workforce. Again, a lot of that is building in redundancy with respect to folks having different skill sets working together to come after that common mission, that common cause, right? So our military stay for 3 to 4 years and rotate in and out and have different subspecialties. It's often microbiologists, it's biochemists, it's veterinarians that do lab animal medicine, it's veterinarians that do research, it's veterinary pathologists, it's laboratorians that focus on clinical medicine, and then a lot of folks that look at aerobiologies. We're focused on an aerobiology threat as a biopathogen, and all those folks coming together, bringing those different skill sets together, where our military kind of travel the world and they see different things and have different skill sets. Our civilian leaders here are immunologists, they're virologists, they're biochemists, they're bacteriologists, which again, they are here not only for their expertise, but for their continuity. John, as an example, has been here 20 years. Jay, I'm not gonna say what year 1980 since you've been here, but these folks have been here for a long time, right? As our military folks come and go with different skill sets, and maybe kind of have a different focus. Our civilian folks are the continuity piece here. They understand what has happened in the past. They understand what's going on in the present and maybe what we should be doing going forward in the future and seeing what has worked in the past and what has not. So that convergence of those two really is a great way to get science done with their different mindsets. And then we also have a contract workforce that is wonderful scientists in here that we bring in to fill different capabilities. They often stay for long periods of time, often end up becoming civilians here eventually if they stay long enough. But it's that flexibility that lets us kind of increase our workforce a little bit with contractors if needed or decrease. And then our military and civilian forces are fairly steady at a kind of a set number, and we can kind of wax and wane with that contractor piece, but it's roughly 1/3, 1/3, 1/3 from the workforce perspective.
So of the, you mentioned to me this morning, there's a military base here with a resident
Correct.
population. They don't all work in the laboratories, correct?
No, they don't. That's a great question. So if we backtrack a little bit, so this installation, right prior to World War II, the Secretary of War kind of did an analysis as we thought that the Axis powers might have bioweapons. We didn't have a program ourselves, offensive or defensive. And so they did that study in 1941 to determine whether or not they were feasible bioweapons, that is, and whether or not we should be looking at that as a country. So then December 7th, 1941, that was Pearl Harbor. This commission, which was a National Academy of Sciences team, they kind of in February of 42 said, yeah, these weapons are feasible, and we being the U.S., should have a program ourselves in the event that we need to combat, our adversaries as we go into World War II to make sure that we are ready for biodefense. And so we, being the United States, then had an offensive program started, and that program was started here. It used to be Camp Detrick. It was an Air National Guard base in 1943. We're about 40-ish miles away from Washington, right? So it seemed like a good fit to be right here. And then over time, the installation increased. More tenants came onto the installation. The National Cancer Institute is here. There are USDA labs here on Fort Detrick, that is. USAMRIID is here. And then shortly after 9/11, it was thought that we wanted a lot of these BSL-4 labs, with USAMRIID kind of being the nidus or the initial BSL-4 tenant. They brought a couple of other labs here as well, the NBAC, the National Biodefense Analysis and Countermeasure Center, which is part of Department of Homeland Security. And then NIH, they have their BSL-4 lab here as well, the Integrated Research Facility. And all three of those are together on this now, went from Camp Detrick to Fort Detrick. And this is kind of this campus, this biodefense campus, wherein as we were walking around, right, we could see the three labs in a horseshoe are very close to one another, with the hopes that will facilitate collaboration and coordination, which largely during Operation Warp Speed, we had a lot of good collaboration. Prior to that, we didn't have as much, we still had some, but that really helped to facilitate that. But that's kind of a brief history on that. And then kind of one aside, the U.S. had an offensive and defensive program until 1969, when Richard Nixon, a second time, the president said, we need to take a look at this, right? Should we really, as a country, have an offensive program? And after that analysis, it came back to the president, he said, yeah, we really, for various reasons, ethical and feasibility and otherwise, maybe the U.S. should not have that program. And he visited here, Fort Detrick in 1969, and basically made a pronouncement that said, we will no longer, as a country, do offensive research. And therein, USAMRIID was born. In 1969, this particular building was built. I think it was finished in 72 maybe. Hopefully the audience can fact check me there. But since then, we've been doing biodefense, right? Creating vaccines, therapeutics, and diagnostics for the warfighter and to protect ourselves. But again, we've had a huge public health benefit to a lot of the things we've done, which I'm sure we'll touch on as a consequence of that. But that's kind of been our mission and a little bit of our history.
So I don't know if you mentioned, but also on this campus is the National Cancer Institute,
Correct. Yep.
which I didn't know. Did you know they were
No.
here? I knew they were in Fort
The
Detrick.
DC area, right?
Yeah.
That's all I knew. I assumed Bethesda.
No, that's far away.
Yeah.
It's really far away. So the viruses you have worked on, Lassa, Machupo, Nipah, Ebola, these are selected because of their relevance to the military. Is that correct?
Correct. Yep. So the military essentially does an analysis and determines what the gaps are. And there's something called the COCOMs, the combatant commands, wherein if you're in the Pacific or you're in the Middle East or you're in South America, and those folks that control those areas, those military commanders say, we have folks deployed in these areas, but we don't have countermeasures for those agents, so we need to develop those. And so as a part of that, then there's funding that comes through the Chem Biodefense Program that is aimed at those gaps. Historically, from a viral perspective, the main two focuses have been filoviruses and alphaviruses. That has expanded and waxed and waned over time, but those are kind of main gaps that we've tried to fill with both diagnostics, vaccines, and therapeutics. But that's kind of the military focus. And so what I've worked on, at least the first time when I was here, going back to filoviruses and alphaviruses, when I got here as a young pathologist in 2010, we had the pathology team here was phenomenal, right? And they were doing a ton of work on filoviruses at the time, and then it continued, as well as alphaviruses. But there wasn't anybody working on these onesie-twosie studies around the edges like Nipah and Lassa and Arena. So I ended up being like the pathologist that did all the other stuff. The other people were doing Ebola and Marburg and alphaviruses, and I got to do hantaviruses, poxviruses, Nipah and Lassa because of that. And a lot of that as a pathologist, you work with the virologists to develop animal models and then test therapeutics. And as a pathologist, you look at things from a systemic level, and I was really fascinated and wanted to look at it at a cellular level as well, hence kind of going back for the PhD.
So if you are making a vaccine or a therapeutic for the military, you still need to go through the normal channels of
Absolutely.
approval, right?
Absolutely.
Testing and approval, FDA and all that, right?
Yep, absolutely.
You have to, and maybe you could get an emergency use authorization or eventually licensure, right? So there's no
Correct.
difference there.
There is no difference there. Now, there's no difference there. That being said, a lot of the things that we work on, like we talked about on the tour, we can get to a certain point, and then short of an outbreak. Then if an outbreak occurs, we can kind of get partnerships and kind of push that forward. But absolutely, everything we use, it needs to go through the same.
So if you developed a Nipah vaccine, you would need to partner with others to do clinical trials, right?
Correct.
How far could you take? Could you do all the preclinical work here?
You could do a lot of the preclinical work here, if not all of it. Yeah. And now again, so some of the pathogens, Nipah, again, some of those things we have. Chem Biodefense has been a great partner and a main funder of us over the years, and some of these other programs we have more or less money depending upon the year and depending upon what their focus is. And so our research efforts in some of these areas tend to wax and wane a little bit. We've had some folks focused on those areas. Sarah Johnston's been a great Nipah virologist here, and Kat Cashman, Lassa, she's done a tremendous amount of work here on Lassa, and other folks in John's lab and in Jay's lab and Crystal's labs, they've done that work as well. But John, do you have anything to add to that? Did I miss anything there?
No, I would just add that a lot of the agents we work with, I don't want to call them orphan agents, but a lot of the time they're neglected as far as having a lot of funding put towards them. So a lot of times we're keeping the back burners lit, kind of keep the research going on those until we need it. And then all of a sudden when you need it, it becomes very important what we've done. There wasn't a lot of research going on in filoviruses and Ebola and Marburg. And then when 2014 hit with West Africa, they started coming to us saying, hey, what do you have for a diagnostic? What do you have for a vaccine? What do you have for a therapeutic? And thank goodness we had actually kept those back burner items going to continue that process, because all of the products that were actually assessed in West Africa in the emergency clinical trials there was a hand that USAMRIID had in those products, whether it was initial discovery and development or whether it was testing down the line. We have a pipeline that allows us to bring in outside products to be tested, as well as the ability to take the basic science from the beginning all the way through to the end. That was a lot more than a second.
Oh, that's great, John.
So we heard today that even though what you work on has a military focus, it spills over into civilian populations, correct? and not just vaccines and antivirals, but therapeutics, diagnostics, not therapy, diagnostic tests, right?
Yes. So basically, we develop the product to protect the service member and their family for when they go into those regions, or if there's a biodefense activity that goes on. But we also realize that everyone that's in the military is also in the global community. It's a general One Health type occupation. So we all hope that what we develop, while it is for the armed service member, makes its way out, and it tends to do that when we come into problems. It makes it way out into the normal populace.
Well, COVID kind of brought all that together, right? Maybe, I don't know if this is the time, but maybe you could
Yes,
maybe
COVID
you could
brought that all together.
describe how COVID hit you. What was your role? What happened?
You want me to take
Go
that?
ahead, John.
Okay. So in 2019, late 2019, early 2020, we all learned of COVID and SARS-CoV-2. When we initially had SARS-CoV-2, it was not a military mission. We were told to stand down until needed. I think that was because we had other missions. We had a chem biodefense mission that we needed to keep going. We needed to continue to work on our diagnostics, our vaccines, and our treatments for Ebola, for Marburg, for Hanta, for all these other agents. And they didn't want to divert us to work on COVID. Then in March of 2020, when COVID basically took over the lexicon of the world, we were redirected and we were told, in addition to your chem biodefense portfolio, we would also like you to do COVID research now.
Who told you that?
The Department of Defense.
Okay.
The Department of Defense asked us to step up our COVID research. Now, interestingly, we had already been laying the groundwork. The virologists and the other institute members here had already had internal meetings to discuss what we would need to do to stand up that type of response. Because we all had an idea that this was not going to end like Zika did like that, and we weren't going to have a problem because of the respiratory nature and the variants that were occurring. So we stood up that capability. Basically, the WashU isolate from Washington State was sent here to USAMRIID first, and at this particular institute, it was aliquoted, and it was analyzed genomically and diagnostically, and we grew up large cultures of that SARS-CoV-2 original variant, which was then provided all across the country and in some foreign countries for them to assess medical countermeasures and develop animal models concurrently with us doing that here at USAMRIID. So we were kind of the repository that provided that virus, so everybody was comparing apples to apples with that early technology. From there, our virologists and our pathologists and our veterinary staff started developing all the animal models that we needed. We put them into every animal model that we had here. We had mice, we had immunocompromised mice, we had hamsters, we had immunocompromised hamsters, we had guinea pigs, we had ferrets, we had 3 species of non-human primates. What am I missing? I think that covers everything. We basically did a very quick analysis. What is the pathology that occurs in those animals? Is it lethal? Most of the cases it was not. But what was it mimicking what we were seeing in humans to bring forward those animal models? And one of those animal models Dr. Hooper will probably speak to, which was the hamster model, which was developed in his laboratory, which was used in Operation Warp Speed. Operation Warp Speed was the US federal response to SARS-CoV-2. And in that, we were asked in a blinded fashion to assess multiple vaccines and multiple therapeutics, and by multiple, I mean hundreds of therapeutics and vaccines in our animal models here at USAMRIID, in combination, as Colonel Bell said, with the IRF, our brothers and sisters at the Integrated Research Facility. So we would test all of those and we would send the data back to the NIH and say, this is what we found, and they would de-blind those studies, and that's what they used to decide how to frontline products for development under Operation Warp Speed. So obviously production, how it could be produced, how much can you make, those kind of components, but also was there efficacy in our animal models. And so that was our major purpose. And then as the COVID response evolved, we continued to assess as the new variants came out. So as a new variant would come out, does the vaccine that we currently have in humans, will it work against that new variant? Does the monoclonal antibody cocktail, does it work against the new variants? Does remdesivir work against the new variants? We continued that process.
So I think most of the public is not aware of all of that. So
Probably not.
hopefully this will help get the word out. I guess Tony Fauci knew what you were up to, right?
Yes, sir. I hope so. Yes.
Now you yourself made some monoclonals, I think, right?
We did, yes.
Building on the work you had been doing with Prometheus. I figured you would have
Yes.
done that, right?
We did. So we partnered with, so the laboratory that I was involved with, so when I ascended to the position of Deputy Director of Foundational, Dr. Andrew Herbert assumed control of the laboratory that I was involved with, the viral immunology branch. And I was involved with the initial discussions. We worked with the company AstraZeneca to develop a monoclonal antibody cocktail that has FDA licensure now. It's called EvuShield. It's given to patients who are immunocompromised because basically the FC effector function has been removed from those antibodies, so you don't have to worry about all those deleterious effects, but it has great binding capabilities and neutralization capabilities. So we combined with them and we used the same technology with Prometheus. So we actually took PBMCs from a SARS-1 patient, not a SARS-CoV-2 patient, and we, through the process of Prometheus, developed those monoclonal antibodies from that survivor of that particular infectious disease.
And this, when you work with a company, how do you benefit from it? Do you benefit? How do you transfer the technology? Because to the world, it looks like the company, GSK in this case, for
It was
AstraZeneca. AstraZeneca, they get all the credit and you don't.
They do. They get a lot of the credit, yes, but we're not in it for the credit. We're in it for the product at the end of the day. So while we partner with these companies, because they provide the capital that you need in order to get these things across the finish line, a lot of the products we develop, we can only take to a certain point. And then we either have to have a catastrophic event, which nobody wants, where companies will come to us and say, what do you have? And then they license it. Or you have a company that's a good Samaritan and wants to bring it forward on its own. Most of the time, it's a catastrophic.
Okay. So you guys never shut down, right? You kept working.
No, sir. As a matter of fact, we actually increased our work. We had our Chem Biodefense mission, and we added on top another 70 to 80% of work on the SARS-CoV-2. To the point where SARS-CoV-2, as you know, is a BSL-3 agent. We actually registered it in our BSL-4 laboratories too. You can always take an agent up in containment level, so we actually doubled or almost tripled our capacity to work on SARS-CoV-2 by registering all of our BSL-4s as well. So we had people that were doing SARS-CoV-2 work in 3s and in 4s at the same time while we were maintaining all of our Chem Biodefense portfolio.
Well, and if I can comment on it too, and I think Rich and I were talking about this yesterday, which is one of the reasons I'm so proud of the team here, is never once did you have to say, nobody complained about coming in, nobody raised an eyebrow about it. It's like, from our folks' perspective, it's like we were, for lack of a better term, kind of an infectious disease war with respect to you just did what you had to do to get the job done, and everybody pulled together and kind of made it happen. And so again, it's just wonderful to see that teamwork like that again from all these different flavors coming together on that singular focus, and to include our brothers and sisters across the street, the IRF, and a lot of the other folks. So again, Rich and I were talking yesterday. A lot of this stuff is not known, but there is a fair amount of collaboration and coordination. It could always be better between the different organizations to make sure that we're doing our best for the American public and the taxpayer to kind of get after.
So I want to circle back to something that John touched on, which is, how is all of this funded?
And everyone's laughing now.
Yeah. So it's kind of roughly split into thirds, shall we say. So the Army gives us operation and maintenance funds every year to kind of maintain the facility and the biosafety, biosecurity, physical security. And then we get money from the Chem Biodefense program, and that generally a pretty big chunk of money. That being said, it does wax and wane over the years depending upon what the priorities are. And then about the other third is us going out and getting after money ourselves, right? So essentially you could look at 2/3 of our funding is essentially soft funding, where we have to compete and go out and get this money, either be it through Chem Biodefense Program, which again, they're a great partner and collaborator, and we generally get roughly a third of our funding from them every year, give or take. And then that other third, we have to go out and get it from BARDA or DARPA or NIH or IARPA or MIDRIP or, name the agency, And then also, too, we have over 500 cooperative research and development agreements, material transfer agreements, interagency agreements, and agreements with AstraZeneca, with Merck, with Pfizer, with all these other folks to make sure that either A, we're working together on something that was started here to get it across the finish line, or B, they have something, they being company X or academic lab Y, that has a BSL-3 or 4 pathogen they need help with, but they don't have the facilities to work on, and they want to bring it here for us to test it for them. And so that's how the work occurs, and that's generally, not to get into too many details, how the funding works.
So is it fair to say that then there's a patchwork of both government and commercial sources, but you have to make proposals to them? Say, we need money to do this.
Yes, and again, I think that, to me, behind the scenes, a lot of our folks that manage that process to keep, John and I, we kind of joke, it's essentially our job to keep the trains running on time. Our scientists are doing amazing stuff, and we don't necessarily do the fun stuff, but we just have to make sure that everybody's moving, and then when somebody comes with a project, that we've got the bandwidth for it, we've got, the equipment for it and the capability for it. But to your point, we've got to have a lot of folks, even in the divisions, that are PMs that are managing all those projects, because there's different colors of money coming from different places on different schedules with different due-outs. And so how do you keep track of all that stuff? So our folks, again, our deputy division chiefs who have been here forever know how to do it, our PMs, those guys are like the unsung heroes here that just keep everything moving. And we are very proud of the fact that we can open up our books and say, this is exactly where this money came from, this is what it was used for, this is when it's expensed, so we can build transparency and trust that there's not some opaque black curtain over us, but we're very transparent in what we do, how we do it, and we're proud to say that we're, again, using the taxpayer dollars in an effective way to get after military health, and then as a second order consequence, public health.
I think most people don't know that. They think that the military budget is huge and that you guys just exist on that you don't have to get your own money. So that's important to get out there.
It is, and that's a kind of a common misconception, I think, that we often have to fight through with respect to getting some of these funders to come work with us. But again, I think, to the Jays, and thank God he's been here since 1987, and John and all these other people, because they have connections and they've got the street cred and they got the clout and they know the folks, and they folks from outside. Sometimes even if it's more expensive to do a study here, people come here because of the John Dyes and the Jay Hoopers, because they trust them, they know their work, and from a military perspective, that comes back to that military mindset. You got to get the job done, and you got to deliver, and you have to deliver on this time frame. And so I think there's value in that teamwork in that regard, because you get that mindset, and then you oftentimes get that output because of that. Again, we make our share of mistakes, and we're not perfect by any stretch of the imagination. But that teamwork like that really does help, I think, get products across the finish line.
I think you had mentioned on our tour that remdesivir was identified as an Ebola virus inhibitor here. Is that correct?
So remdesivir was a product from Gilead. It's a well-known product now. And it was screened by Gilead for a litany of viruses, but not the viruses that we work on. So they contacted us to ask us if we would screen against our panel of BSAT agents, and Marburg and Ebola and filoviruses were very prominent on that hit list. And then Gilead has a cooperative agreement with us, and we're working with Gilead first for Ebola virus and now for Marburg virus to bring it forward under the FDA animal rule. So that's a real winning situation. And then remdesivir obviously was also used for COVID as well. So it's a nice broadly active, broad spectrum antiviral.
Is it used in Ebola cases at all?
So it was used in the PALM trials in the DRC, and it did show positive effects in that people received it, but it was not as good as one of the monoclonal antibodies that was given. So that product has been moved forward for Ebola, and now they're repurposing it for Marburg.
So Jay, hamsters were mentioned a little bit, and you developed this hamster model for COVID. So tell us how that came about.
We use hamsters for a lot of our research. Mostly for me, it was hantavirus research. And the literature for SARS-1, they had put SARS-1 in, and they didn't really They didn't get good disease, but there was infection, and it looked like something to try. So, I mean, we literally had hamsters here. We were using them. We repurposed them. As soon as we got that Washington 1 strain, we went in and saw the weight loss and the lung pathology. So it looked like a good model. But to really confirm a model, you want to test out a medical countermeasure and make sure that disease is caused by the agent, the virus in this case. And so we have a DNA vaccine capability. So we were able to, as soon as that sequence came out, put in an order to have the gene synthesized, to pop it into our platform. We were also working with a company that had done, like what John said, had converted a SARS-1 monoclonal. And so we were able to test that in our hamster model, show that we could get protection with the DNA vaccine or with the monoclonal. And we took it a step further because our hantavirus research, we had found that when you transiently immunosuppress a hamster, you can get more intense disease. And so we used the same technique, which is basically treating with cyclophosphamide and dexamethasone, treated the hamsters after being vaccinated, and you get dramatic weight loss and sustained weight loss in the control animals, whereas the vaccinated animals or the antibodies, the animals treated with the monoclonal were protected. So we did all that, and I'll do a shout out to Joe Golden out here, because what we also did is we have connections with the facilities that produce transgenic animals, like Jackson Labs, and so we were I think we might have been the first lab to get the ACE2 mice, human ACE2 transgenic mice, and so we got that model going up right about the same time Michael Diamond did. And so we were able to test medical countermeasures in the mouse model also. So we did that all early on, very early on, before the field kind of overtook us and swamped us out. So,
So did you say one of your the medical countermeasures you tested in the hamsters was a monoclonal? So where'd the monoclonal? This is all happening really fast. Where'd the monoclonal come from?
That monoclonal antibody that we tested very early was actually from a company called Centivax, and they had converted a SARS-1 monoclonal antibody by changing a few amino acids, and it worked. But then as the variant started appearing, we were able to pop those genes into our DNA vaccine, and we actually make pseudovariants with the DNA vaccine also, so VSV pseudovariants, and so we were able to look at neutralizing antibodies to the different variants as they emerged. And so it was a running battle for a while, where it seemed like every couple weeks there was a new variant that we were popping into our system and measuring cross-neutralizing antibodies. And unfortunately, a lot of the antibodies that we tested as part of Operation Warp Speed were overcome by the variants and no longer neutralized. So, that's how we worked with the hamsters early on.
So this may be a bit of a sidetrack, but I'm interested in the DNA vaccines, because you have a long experience in this, right?
Yeah.
I'm interested in your two things. How do you deliver the DNA vaccine, number one, and number two, your spin on the comparison between now DNA and RNA as a platform for vaccine delivery.
Yeah. So delivery of the DNA is the hard part. That's the big challenge. And if you go needle injection, it really doesn't work. So needle and syringe, a lot of DNA vaccines, HIV DNA vaccines, they were trying needle and syringe and said, hey, this isn't working. And so then the technologies evolved to like gene gun, which is putting the DNA on gold beads and shooting those into the skin. And that works really well, but it's kind of cumbersome. We got up through we actually did, I think the last vaccine phase 1 trial at USAMRIID was our hantavirus DNA vaccine delivered using the gene gun. And at that time, Pfizer had developed a kind of a all-in-one, so that basically was a device that was manufactured with the DNA as a powder, gold powder in the device. But you can imagine storing those things. It's like a device. It's stability was a pain, and so Pfizer kind of dropped out. So then electroporation, we moved forward along with the companies that had electroporation systems. So electroporation basically opens up the cells, it lets DNA in at the injection site more efficiently. But again, it's cumbersome, it can be painful, kind of scary when you see all the needles involved. And so then there had been another method called jet injection, and it was pretty clunky. There was it's called biojectors, and they would shoot the DNA in as basically a liquid
Is this
needle.
the same sort of thing that was used for smallpox?
That's what everybody thinks. It might have been similar to that type of jet injection. Actually, I think it was, but that wasn't necessarily a needle injection. That was more of a kind of mimicking the bifurcated needle scarification,
okay.
only over a larger patch of skin. Anyways, a company called PharmaJet developed a jet injection technology for use in conventional vaccines, and it was designed to use in resource poor countries, that kind of thing. So it's very cheap. It's disposable syringes that you hook onto this little device that you it's spring powered, and it's jet injection. So we tested that with a DNA vaccine and found a significant increase in immunogenicity after using that in rabbits and non-human primates. And so then we moved, so we have a DNA vaccine for a couple of actually multiple hantaviruses. And we had already gone into that phase 1 with gene gun. We had gone into phase 1 and 2 with electroporation. So now we've gone in with a jet injection into a phase 1, and right now we're in a phase 2. And we get great neutralizing antibody responses. It protects in animal models. And you asked earlier about moving this is an in-house product basically, and we've gotten it through phase 1. We're in phase 2, but the problem is, what do we do from there? John talked about outbreaks and things like that basically stimulate funding and interest from industry. Well, what if you don't have that? What if it's a threat for the military, but you don't have the commercial incentive? That's where we are right now. It's difficult, and you might know about the FDA voucher program, where it's a program that's trying to incentivize companies to work on orphan indications. But those all cover tropical diseases and some biodefense, but not hantaviruses. So we are falling right between biodefense and public health. It's military kind of specific, but we'll figure it out. And so that is a challenge.
So the delivery by a gun of some kind is a military specific delivery, right? It wouldn't be used for the general population.
No, it would be used.
Yeah.
So the PharmaJet devices, they were developed, and they are being used for polio eradication campaigns, like in Pakistan.
Kids are okay with these guns, yeah.
Oh yeah.
They don't have a problem.
They have it doesn't look like a gun. It looks like
a bottle of water.
Yeah, and you basically push a button, and there's an intradermal device and a intramuscular. Polio is intradermal. The kids don't even, you can go on the PharmaJet website, watch the videos. The kids don't even know they're being vaccinated. Now with the intramuscular device, you know you're being vaccinated. It feels like a rubber band's kind of snapping into your arm.
Why would that work and an injection not?
Good question. What I think it is, it's the instantaneous, the pressure over milliseconds that's blowing a volume of fluid into a tissue. And so by physics, it's the amount of DNA that's actually getting into cells is increased. Now we've also looked at lipid nanoparticle delivery.
I was going to ask.
With DNA, and boy, it jacks it up. The problem is we haven't worked out toxicity issues. Now with the messenger RNA, with lipid nanoparticles, they worked out the toxicity issues, right? They're right kind of on the edge, but they worked it out. So yeah, how does messenger RNA compare with DNA? What messenger RNA has a huge infrastructure, industrial infrastructure built right now. DNA really doesn't. There's companies that manufacture the DNA, but that's it. So that's the huge advantage of messenger RNA. That the back end is just really, really interesting. But you can take DNA vaccines and easily convert them to messenger RNA vaccines because you've discovered, you know the target, you have all the assays, you've tested in animals. It's very, very easy to transition over, and we should probably do that. So,
So you published about using mRNAs encoding antibodies as a way of making people make their own antibodies. Tell us about that.
yeah, so this was a DARPA funded project. Eric Mecker was involved. And basically what this project's goal was to be able to take nucleic acid, so DNA or RNA, encoding the genes that encode the heavy and light chains of a monoclonal antibody, deliver that DNA or RNA into an animal. And the idea is that then the monoclonal antibody proteins are produced in the cells of the host and find their way into the serum. And so you basically have turned, say, a person. If you're vaccinated with this, they now become the bioreactor. They're producing the antibody rather than being injected with the antibody. And so we got as far as showing that we could do that with messenger RNA, LNP formulated messenger RNA, or naked DNA, or LNP formulated DNA delivered by jet injection. The model we were using was rabbits. The antibody that we were launching was a neutralizing antibody against smallpox, pox viruses. Yeah, it was very exciting. There are companies still working on that, and I think that will become a technology that will be used to basically get antibodies produced without needing to produce the antibody in bioreactors. You're basically just making nucleic acid using the exact same technology that these messenger RNA companies are already using to deliver antibody genes.
Yeah, so that's, in your view, that's the advantage. Instead of having to produce large quantities of monoclonals under the right manufacturing conditions, you just make the mRNA.
Yep, so you can use the same technology that you're using to make vaccines. You might want to try and do what you can to decrease the adjuvant effect of the vaccine, because you do not want a response to those human antibodies that you're making. And that's one of the problems that I think we ran into. We were making human antibodies in rabbits. And so the rabbit's immune system kicks in and starts taking those out of circulation. But the idea is that in humans, you'd be using a human antibody, and it would be almost, you would have detectable levels within a day or two.
So this brings up something that struck me from one of your bios, the transchromosomal cow?
Yeah. So
what is that?
yeah. So again, we'll do whatever we can to make a medical countermeasure to one of our targets. So again, hantaviruses. We had our DNA vaccines. We knew that when we vaccinated monkeys or other ant rabbits, that the antibody produced, we could go into our hamster models and show we could protect. Well, what if those antibodies were human, those polyclonal antibodies, instead of rabbit or monkey? So this company, it's called SAB Biotherapeutics. They engineered these cows that they basically knocked out the bovine genes that encode the antibody, all of the antibodies, and replaced that with human genes, basically pieces of human chromosome encoding the human genes for antibodies. And over a series of engineering basically accomplishments, they were able to get animals that now produce fully human antibodies. And so what we did is we took our DNA vaccines, vaccinated these animals at SAB Bio. They have a GMP capability to make plasma, purify out the antibodies. They come back to us. We use our live virus neutralization assays or our pseudovariant neutralization assays. We show that there's neutralizing activity, then we go into the animal models. So it's super potent. The neutralizing antibodies in these cows, the titers are up over 100,000. And we've, through an R01 grant, we were able to make a pan-hantavirus polyclonal antibody by vaccinating with 4 different hantavirus DNA vaccines. It's gone through GMP safety testing. It's gone through human tissue binding safety testing. So it's a polyclonal antibody. It looks very potent in the protection models. The problem is the market. We're hitting a brick wall. So, but yeah, these transchromosomal bovine have been used for, you don't have to vaccinate with DNA. You can vaccinate with whatever vaccine you want to. It's been used for Ebola, for SARS. They have a product called SAB-185 that is variant proof to date. That's a nice thing about polyclonal. So
Why cows? Because they're big and you know how to take care of them?
They're big. They're big, and it's mostly the size.
Yeah. Okay.
This company also spun out another company called Capravax, and that's goats.
Goats.
Yeah.
Yeah. We know how to deal with cows.
Yeah.
Right?
Right.
Lots of cows in the U.S. All right, Crystal, let's talk with you a bit. Sorry,
No, it's fine.
we got into these cows here. Tell us about your equine encephalitis viruses. Why are you interested in them? Is this a military interest?
So, in general, for USAMRIID, yes, it's a military interest. When the offensive program was happening many, many moons ago, in the 40s, not only did USAMRIID experiment with the alpha viruses, but the former USSR did as well. And so since those times, the alpha viruses have always been on the military radar as a potential threat agent. And surprisingly, for being small RNA virus, but maybe really not now that we know so much more, they've been really difficult to get good countermeasures against. Out of all of the alpha viruses for the military, Venezuelan equine encephalitis virus is probably the most important.
Why is that?
Mostly because there are multiple subtypes. So the genetic variants in the glycoproteins make it very difficult to get a specific medical countermeasure to work against all the different varieties. Kind of like a SARS-CoV-2 problem, right? You come up with something, but there's always another one out there that can get around it. For the alpha viruses, what the strategy seems to be right now is we really want something that works against the 3 main encephalitic ones. So Eastern, which is a problem in the United States, Western, which honestly hasn't really been seen since 1990, and then V, which as we know is endemic down in Central and South America. When I first got here, Dr. Glass had a program or project with NIAID to test their vaccine or virus-like particle platform against all 3 of the viruses. And actually we tested them in mice and in non-human primates, giving the trivalent formulation all at the same time, and it works beautifully. But much like you've heard for the hantaviruses, you can get it to a certain place, but unless there's a catastrophic event, it's hard to get past that point. And so we've tested it up to pre-phase 1. Then NIAID licensed it to Emergent, think NIAID actually did phase 1 and then licensed it to Emergent. And so on and off over the past few years, we've had small projects with Emergent to try to continue getting the necessary data packet to go to the FDA with it. But unfortunately, we're still not there.
So the idea would be that you have it now through phase 1. So if there's an outbreak, you could then do a phase 2, right?
Correct. Yeah. Another large thing that we've been doing over the past 10 years is to develop animal models, appropriate, I should say, animal models for the encephalitic alphaviruses. Because in the US, we may get 30 infections a year that we know about. Usually they're fairly fatal, with over 50 to 70% of those who make it to the clinic end up succumbing. It doesn't give you a large population to do these trials on. So to have an appropriate animal model that has a similar disease to what you would see in humans is necessary to be able to take to the FDA with any medical countermeasure. And so we've spent a lot of time developing non-human primate models for Venezuelan mostly, but also Eastern.
So you say these are empty particles, right? Virus-like particles. You just produce the structural proteins, right?
Much, yeah. It's pretty much anything after the 26S promoter of the alphaviruses. So there's 2 promoters on those genomes, and you take the structural part and put it into an expression vector, and you can make very pretty particles that look just like a virus, but have nothing inside of them.
And you can purify those for, do you need an adjuvant?
We have done adjuvant testing, and if you use, we used alhydrogel for the one test we did, you can reduce from having to give 6 micrograms of protein, which really isn't that much, all the way down to 0.6 micrograms of protein. Wow. So, adjuvanted, it's even more potent.
What do you use for vectors to produce the proteins? A variety of things, or first
they were using a CMV vector, I think.
Okay.
I'm sure you could probably use a variety of different vectors to express. It's just the important part is having all of the right parts of the structural proteins in there to get it to fold properly.
So then if you take these virus-like particles and immunize an animal, I don't know what animals you've used.
We've done mice and cynomolgus macaques.
Okay. Then you challenge them, they're protected against disease. Okay, and do you know if it's antibodies or T cells or both that does that?
So the thought in the field is mostly that it's the antibody response. However, that being said, we do see in mouse studies where you can have larger numbers that even animals without a neutralizing antibody response do have protection. So it could be the non-neutralizing antibodies present or a T or B cell response. And there's really just hasn't been an interest in diving into that mechanism of protection. So as long as it protects, we're good.
Sure. So you've also made monoclonals against these viruses, right? For therapeutic purposes?
Yeah, so we had a project when I first got here to actually look at monoclonals developed many moons ago by the CDC and to express them via a plant and isolate them and then test. 2 of them made it to the non-human primate model. Both provided some protection. 1A3B7 actually was less potent of a neutralizer, but a better binder and provided better protection than the more neutralizing antibody. So I guess that's a plug for don't discount non-neutralizers, they can be important. But also we have a more recent project working with the Navy Research Lab down in DC. They are using single domain antibodies. So these little camelid antibodies. We got a bunch of sera from 3 llamas, which was fun because they got labeled Centavo, Whisper, and Cowboy. And I was like, all right, we're testing Cowboy today, y'all. But we've got sera from vaccinated llamas. We tested them against a variety of viruses, told them basically, Whisper was the best. They then did phage display from those libraries, picked out a variety of different options, sent them back to us. We screened them, and we recently tested them in vivo in mice and did get protection. So pretty excited about that, and I'm hoping that project ended in the fall, and we're hoping to get funding to continue it this coming fall. So fingers crossed that someone sees some benefit to the single domain antibodies. Originally, we started looking at those because they're smaller, and with encephalitic viruses, the idea is, you might get some antibody that could cross into the CNS and provide protection. That is yet to be proven, but that is something that we would like to look at in the future.
All of you have said you wanted to work on something that made a difference, but it's hard because when you run up against these walls, when there's no disease to justify the next step, it's not like Rich or I, when we had our labs, we could do some interesting project, right, that we thought was cool. May not help anyone, but you can't do this. So what do?
You go where the mission takes you.
Yeah.
You put it in your side pocket for when the opportunity comes, and you be like, oh, look at this really cool thing that we've been waiting on for someone to be interested in. And then you move on, because eventually they do come back to it.
There's probably more to do than you have time or personnel for or money for anyway, right? So there's always something.
Did you get pulled into the COVID effort at all?
I was one of the people that was maintained mostly on CBD, chem biodefense purposes. I did assist in some ferret influenza, SARS-CoV-2, not transmission, but co-infection studies. But that was the most that I was involved in that part.
Okay.
So a question that's coming to me that's relevant not just to your mission, but sort of epidemic preparedness in general is, you've talked about being able to get up to or maybe through phase 1, and then you can't get beyond that because the interest wanes, okay? Is that not good enough? All right? And how, I suppose it's not good enough from the point of view that if something really hits and people are dying right and left, you don't want to have to spend the time on phase 2. And somebody must be doing this calculation, okay, as to how ready is ready enough. And, do you have an answer for that?
I can try an answer for that. So there's schools of thought. You can spend a lot of money and a lot of energy and a lot of effort taking something through the animal rule with the FDA all the way through to licensure. Or you can fill the freezer with 10 to 20 to 30 to 40 different products that are up to the phase 1 with GMP product ready to go, so you're prepared for that. It depends on your mission. If you're talking about a global health mission, and you're talking about a non-pandemic, non-catastrophic event, maybe the latter is okay, because you have 50 different products against 50 different agents that you're able to pull that out of the freezer and move it very quickly through an emergency use clinical study. For our military mission, some people look at it a little differently. We have a select group of agents that we are interested in from a military perspective that we know an adversary may choose to use. So therefore, having something that's more advanced down that pathway may be preferable. I am not sold on either pathway. I think there's probably a mixture of both that is needed, depending on what your mission is and who you work for. As we have moved into this century, we have learned that the biodefense community and emerging infectious disease community are now interwoven and indistinguishable. Anything that you have endemic could be potentially used as a bioweapon, and vice versa. Anything that's a bioweapon could potentially become endemic, and we've seen that potentially happen. That being said, how are we prepared? How are we best prepared? I assume there's some bean counters somewhere, some actuaries that are actually running the numbers to know where our money is better spent. In the meantime, as Dr. Burke said and Dr. Hooper said, our job is to keep the research moving as best we can over time, putting things on the back burner of the stove until we can actually move it to the front burner and turn it on, because at the end of the day, they always come back. These aren't going away. most of the time these emerging infectious diseases are re-emerging infectious diseases, or a brother or sister or cousin of a previous virus or bacteria that we've seen before. So we just try to be, I think Colonel Bell said it at the very beginning, we have to be prepared. Scout motto, be prepared, to be able to handle these as best we can.
So the military mission can that be divided into protecting soldiers going into areas where there may be endemic pathogens, okay, that they would encounter on the one hand, and on the other hand, biodefense against somebody producing a bioweapon? Where do the alpha viruses fall into that?
Depends on which alpha virus you talk about.
Okay. Some of them you're talking about going into endemic areas, others potentially bioweapons.
Yeah, so in recent years with chikungunya becoming such widespread, it's become, on the forefront of CBD's mind and is actually popping up more in the request for proposals. They want people looking at chikungunya therapeutics and vaccines and things like that. 5 years ago, it wasn't even on their radar. It's an alpha virus, so we know we have the expertise, we can do it, but the models are quite different because it's a very different disease.
Weren't the smallpox antivirals and vaccine stockpiles made in case of a
That was bioweapons,
and
yeah.
which had a collateral effect in the mpox outbreak, right?
Good point.
We used them, so you never
I was
know.
wondering about in this scenario, mission scenario, where do the hantaviruses fit in?
Those are not considered bioweapons by the DoD. They are of military importance because the troops run into them. They're carried by rodents. The troops run into them in endemic areas. But they're also a Category A agent for NIAID. So NIAID considers it a possible biological weapons threat. So, but with the DoD, it's the unnaturally occurring threat. But I'll just comment, like what John said, that's the idea of stockpiling and being ready is, I think, the way to go for antibodies, small molecule drugs. But for vaccines, vaccines work best when you're vaccinated, when you're protected, especially for the military, for an attack. You want to be vaccinated. You don't want to come in after the fact. And so the need to go a little bit further for vaccines, I think, is greater, but they're often treated the same way as an antibody or a small molecule. So, and sometimes the plan is to use the vaccine after the fact, but, and that might mitigate the problem, but it might not. So.
All right, here's a question. Are there any military-specific vaccines that other people don't get, not civilians don't get? Anybody know? Adenovirus is one, yeah.
Yeah.
Any others? That's the only one I can think of.
Then, anthrax and smallpox prior to
That's true.
some deployments,
Yeah.
those were given
Yeah.
out.
And also in our institute, we had an SIP, Special Immunizations Program, where there were certain agents that we would work with on a daily basis. So for instance, I've been vaccinated against Western, Eastern, and Venezuelan equine encephalitis virus.
With what?
They're
Investigational
They're
vaccines.
IND
Yeah,
Okay.
so there were two, the Western and Eastern were formalin inactivated preparations, and then the V was TC83, which you've probably seen if you look at the literature and people who use it as a surrogate for V. The problem with all of them is they're not that good. And
You hear
for
that,
V,
John?
people,
I'll tell you, that V kicked my butt.
yeah, that is
That
why
V
it
vaccine,
never
I
went.
felt like elephants were
So that's
trampling me in my head.
An
So
attenuated.
the TC83 had a high reversion rate, so it was
Okay.
never beyond IND.
I have two more questions, just so you know we have the end in sight, folks. I'm sorry. First, what is an advantage of doing research with the military, Todd?
That's a great question. I think there's an advantage with respect to, at least here, the things that you get to work with and the people you get to work with as an association with that, right? So again, when I came here in 2000, had I wanted to work with Nipah or Ebola or Lassa, one of the only places where you could do that would be here. And that was, something that I was interested in. I found out over time. And so I think that is really key, combined with the fact that, from a military perspective, it's just, it brings a unique collection of people together, which is really cool because, the military is a slice of the United States coming from, it's kind of a melting pot. And so getting to work with all these different people that have different backgrounds is really pretty impressive.
About you, Jay?
Yeah, after being here and then going into hardcore academia, being here and having the mission and thinking that, we're trying to protect the military, and by doing that, protecting our families, the country, just having that dynamic to the research, I really like. And a lot of the things that we work with are very international. It's very, these diseases are happening all over the world, so it's kind of a global thing too. So it's
Got it.
that's what I like.
Yeah. Did you ever have a mission, Rich?
No, and and that's what really impresses me about this. I mean, the mission is, it's ever present here, and I'm really impressed. I really like it.
Crystal?
I was drawn here to mission, right? Feeling a purpose of why you're doing your research. It brings every day, even if it's the worst day, you're here for a reason. And I've worked at a couple of different places, and the people at USAMRIID are amazing. They're what keeps it going, the collaborative spirit, the drive to get it done, not just because we want to do it, but because people are relying on us to do it. It makes it worthwhile.
John?
So they stole two of mine, mission and global aspect. I've been able to travel all over the world and meet people from different cultures and talk to them and help them with their science and learn from them. I think that's an amazing opportunity that I would never have had if I didn't come here. The two other things that they didn't talk about, number one, I get to come to work every day and side-by-side work with the soldiers that I'm trying to protect, that I'm working with them to protect. That provides gravitas to what we do. Every day we look around and we see why we're here and why it's important we're here. And the last thing is, USAMRIID is a magical place. We've heard that before. The magical kingdom, I'm gonna get my Mickey Mouse ears. But it's a magical place because you can take something from basic science discovery all the way through to advanced development into a product. And I don't know of any other place in the world where you can do that. People have so specialized in particular areas of research, where you do clinical studies or you do the basic science or you do preclinical. We literally have that capability here with the people in this room to be able to do that from end to end, soup to nuts, a product. And we also have the ability to siphon in other products from other people. I think that's an amazing place to work. And as a scientist, I can't imagine a better place to work.
All right, one more. Todd, if you hadn't been a scientist, what would you have done with your life?
Oh, good question. Well, I knew I had to be a scientist. I grew up on a farm, and every time my dad left, I would break something, and I was not talented enough to be a farmer, so I couldn't have been a farmer. Because they were economists and agronomists and animal scientists. They were multi-talented. But I always thought I was going to be the shortstop for the Chicago Cubs until I was 40, and then I was going to be a vet. So had I had the athletic ability and the height, I probably would have been a shortstop for the Cubs.
Are you right-handed?
I am.
Okay.
So I need to teach my son how to throw left-handed.
Jay?
Boy, that's a tough one.
I know.
It's a tough one. I think I was really interested in the idea of biotech, kind of business. And so I thought maybe I would go that way, but it would be somehow connected to science. So if I couldn't do that, boy, I don't know.
Well, business is okay. That's an answer. Okay.
There you go.
You got it. Crystal?
So I originally was wanted to be a veterinarian, so it's still science related, but if I wasn't doing either of those things, I probably would have worked at like the national park system or a conservation center or something like that, because I grew up not on a farm, not Iowa style, but I had 2 horses and a bunch of dogs and cats, and I always loved my animals, and so I probably would have done something that related to ecology and animal health.
Okay. And John?
I have two answers. The first, the serious answer would be, I always thought I wanted to be a marine biologist. And then I discovered I was seasick. I'm not kidding. So that really ended that possibility. So then I decided to move into more biology. And then honestly, when I was growing up, I always wanted to be a professional basketball player. But I learned very quickly that there are a lot taller and a lot better and a lot more skilled individuals than myself out there on the basketball court. So that ended in high school, and that was about the end of that.
You're probably still a good basketball player, right?
I'm okay. I can hold my own.
All right, that is TWiV 1015. You can find show notes at microbe.tv/TWiV, and you can send questions and comments to twiv@microbe.tv. And if you love what we do, even if you like it, we'd love to have your financial support. You can go over to microbe.tv/contribute. Microbe TV is a 501(c) nonprofit. We exist on philanthropy. We don't do ads on our program, so we need your support. Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Always a pleasure. Thanks very much. And thank everybody here. This is great stuff.
And thank you guys for coming today. It's been a real pleasure and honor to have you guys here, especially on the celebrating or commemorating D-Day, and just coming here to understand what USAMRIID is all about and display some of the myths out there. And we're always looking for some additional great young men and women to work here. So out there, if you're interested in USAMRIID, please look us up. We'd love to have you come to USAMRIID and visit and work with us.
How would they look you up? What would they do?
So there's NRC postdoc opportunities that are published, ORISE opportunities, and you can go to our website. There's ways that you can contact us and find out what employment opportunities there are here, as well as collaborations. Our research program office, led by Colonel Swarczewski, is a great point of contact, which can also be found on our website. So if you're a company or a collaborator out there that wants to work with us, go to our website, check that out, or get a hold of myself or John, and we'll be happy to tie you in. So thank you.
Our guest today from USAMRIID, Todd Bell, thank you for coming and also for initiating this a year ago.
Oh, no, thank you again, guys, for coming. It's been a real pleasure.
Jay Hooper, thanks very much.
Thank you.
Crystal Burke, thank you.
Thank you, too.
And John Dye, thanks very much.
Thank you, sir.
I'm Vincent Racaniello. There goes that sir again. Yeah. You can find me at virology.ws. I'd like to thank the American Society for Virology, the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.